Hello students, I am Mohan Kumari. Welcome back to MK Classes for Biology. Yes, in today's class, we'll be learning about another important and amazing process in the chapter Human Reproduction, that is fertilization. Yes, we are going to study about fertilization in today's video. So before going to start with this concept, those who are watching my video, please like, share and don't forget to subscribe. Yes, here we begin with the process of fertilization. As I told you before, fertilization, it is the fusion of haploid sperm and ovum to form diploid zygote. Before fertilization, there are certain events which are necessary. That includes release of ovum from the ovary into the fallopian tube. So release of sperm into the female genital tract in order to reach the fallopian tube, that is the site for fertilization. Let us study these two events now. That is the release of ovum first. Now, rapid secretion of LH leading to its maximum level during the midday of the menstrual cycle called LH surge, that is called as the ovulatory phase of the menstrual cycle. Due to LH surge, induces the rupture of Graafian follicle and thereby the release of ovum by the ovary takes place. Ovum here is the secondary oocyte, which is transported to the ampullary isthmic junction of the fallopian tube where fertilization takes place. Yes. Once the Graafian follicle rupture and releases the secondary oocyte from the ovary, that secondary oocyte received by the fimbriae of the infundibulum, finger-like structures present at the tip of the infundibulum, that receives the secondary oocyte and transported to the received secondary oocyte transported to the ampullary isthmic junction of the fallopian tube where fertilization takes place by the ciliary movement of the fallopian tube. So second is the release of sperm. So during intercourse or the coitus, semen is released into the vagina, which consists of sperms and seminal fluid. This process is called as the insemination. The motile sperms, which swim rapidly and passes the cervix and enter into the uterus and finally reach the junction of the isthmus and ampulla of the fallopian tube, that is site for fertilization. Now, fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary isthmic junction. This is the reason why all copulation do not lead to fertilization and pregnancy. So there is a phenomena called capacitation. Once the sperm enters the female genital tract, it should show capacitation. That is nothing but the process of sperm activation within the female genital tract, so that then only the sperm can reach the egg. After the release of gametes into the fallopian tube, now the process of fertilization takes place. Fertilization is the process of fusion of sperm with an ovum. So during fertilization, a sperm comes in contact with the Zona pellucida layer of the ovum, which is said to be the middle layer of the ovum, and induces changes in the membrane that block the entry of additional sperms. Yes. When one sperm reaches the Zona pellucida, it will bind to the Zona pellucida because of the receptors which helps in binding of sperm to the Zona pellucida layer. So this creates or induces changes in that membrane, the membrane become hard, that block the entry of additional sperms. Now you can say that is the zona pellucida layer. Thus, it ensures that only one sperm can penetrate into the egg and fertilize the egg. So this is called as monospermy. This is extra information for you in order to understand how the sperm is going to get attached with the zona pellucida layer of the secondary oocyte and how it is going to cross that zona pellucida and reaches the inner membrane of the egg, and what are the changes you can see when the sperm head attaches with the zona pellucida in order to achieve monospermy and prevent polyspermy, and how the sperm nucleus enter into the cytoplasm of the ovum. In order to explain these process, I am going to explain in 3 sub processes, that is acrosome reaction, cortical reaction and fertilization cone. Let us see what are these. So the first one is acrosome reaction. So now you can see zona pellucida of the secondary oocyte, that is the middle layer of the secondary oocyte. The outermost layer is the corona radiata. The sperm can pass through this corona radiata but cannot cross the zona pellucida. So, zona pellucida of the secondary oocyte in the fallopian tube consists of ZP3 receptor proteins. It is a glycoprotein. ZP3 receptor function as sperm receptor. This and bind with the complementary molecule on the surface of the sperm head. So you can see ZP3 receptor present in the zona pellucida membrane. It functions as sperm receptor. So because of this receptor, the sperm consists of complementary molecule to this receptor that facilitate the attachment of this sperm to the zona pellucida layer. Once it attached with the zona pellucida layer, that induces acrosome to release hydrolytic enzyme. We had studied the structure of sperm earlier. The tip of the head region of the sperm consists of the region called acrosome. This acrosome enclosing the hydrolytic enzyme. When the sperm head attaches to the zona pellucida layer, due to the ZP3 receptor and the sperm is having complementary molecule for the receptor, so that induces acrosome to release hydrolytic enzyme in order to dissolve that particular region of the zona pellucida. The secretion of acrosome help the sperm to cross the zona pellucida and get fused with the inner layer, that is the plasma membrane of the secondary oocyte. Now the sperm has crossed the zona pellucida and it is attached with the oolemma of the secondary oocyte. You can see here the second reaction is the cortical reaction. How the cortical reaction helps to prevent polyspermy, we are going to study. Now during the fusion of sperm and egg plasma membrane, in the previous acrosome reaction we had studied, sperm has already crossed the zona pellucida layer by dissolving the particular region in the Zona pellucida due to the hydrolytic enzyme. Now it has crossed the zona pellucida and attaches the plasma membrane. During the fusion of sperm and the egg plasma membrane, egg now show cortical reaction. What is that cortical reaction? In order to prevent polyspermy. Just you can see here, the cortical granules present beneath the egg plasma membrane. You can see here, these are the cortical granules which are present beneath the egg plasma membrane are released their contents between plasma membrane and the zona pellucida. So inner membrane is the plasma membrane. Next to plasma membrane is the zona pellucida. In between these two membranes there is a space called perivitelin space. Now these cortical granules which are present beneath the plasma membrane are release their contents into the perivitelin space, that is between the plasma membrane and the zona pellucida. So because of this secretion, you can see that is the cortical granules release the secretion or the contents into the perivitelin space. When the enzymes which are released by the cortical cells into the perivitelin space, next to perivitelin is the zona pellucida. Because of this enzymatic reaction, zona pellucida get hard. So the enzyme released by the cortical cells harden the zona pellucida to prevent polyspermy. Once the zona pellucida become hard, so now next sperm cannot enter or pass the zona pellucida. This is how polyspermy can be prevented. That is about the cortical reaction. After this cortical reaction, our sperm which is there out of the oolemma, that is plasma membrane of the secondary oocyte. Now the fertilization cone is going to form. That means when the sperm penetrates into the egg produces, can you see here, egg produces fertilization cone. So egg is going to form a fertilization cone into which the head and middle piece of the sperm get engulfed. So this is how the sperm enter into the cytoplasm of the egg through the plasma membrane of the egg. So the sperm entry into the secondary oocyte, which induces the completion of the meiotic division of the secondary oocyte. The second meiotic division is also unequal. We had studied this in oogenesis, and result in the formation of a second polar body which undergo degeneration and the haploid ovum. Now later the haploid nucleus of the sperm and that of ovum is fused together to form diploid zygote. This is how you can see the process of fertilization, which is very beautiful and amazing process. So there is a concept called sex determination of a child. How can we determine the sex of the child? So the sex of the child is determined at the stage of fertilization. The sex chromosomes of the male parent are X and Y. So human male consists of allosomes of two types, that is X and Y, which determine the sex of that particular individual. Hence, male individual produces two types of sperms carrying X or Y chromosomes. So male can produce sperms. So those sperms can carry either X or Y chromosomes. Hence, male is heterogametic. And that means the male can produce sperms. Sperms are of same type. But if we see the chromosome, sex chromosome in that sperm, the 50% of the sperm consists of X chromosome and the 50% of the sperm consists of Y chromosome. Whereas the female parent has XX chromosomes. In case of human female, allosomes are of single type, that is X type. A pair of allosomes, both are of same type, that is XX. And she can produce only one type of ovum, all the eggs produced by the female carrying X chromosome only. So, female is called homogametic. That means all the gametes produced by the female individual carrying only X chromosome. After the fusion of sperm and ovum, the zygote would carry either XX or XY chromosomes. Now you can say the male sperms of two types, 50% of X chromosome carrying X chromosome and 50% carrying Y chromosomes and Y chromosome, and all the eggs carrying only X chromosome as allosome. So now when this X chromosome carrying sperm fuses with the egg always carrying X chromosome only, fuses with the egg, it is going to form a diploid zygote carrying both XX chromosomes. Now these individual, which is going to develop as female child. If the sperm carrying Y chromosome, when it fuses with the egg carrying X chromosome, the resultant offspring is going to form is XY. So these individuals which are going to develop as male individuals or male child. This is how sex determination can be studied in humans. So now you can say XX zygote develop into female child, XY zygote develop into male child. Thank you. So this is about today's video. Do watch my video. Thank you.